All right, folks, 18-2 is really the meat of the chapter, and so this is going to be a little bit of a longer lecture. So this is it right here, and then when we do 18-3 and on, it gets a little easier, but this is the meat of it. All right, so first of all, we have three classes of hormones. The first classes of hormone you see here are what we call amino acid derivatives, and these are water soluble for the most part. Actually, the exception to that is that right here. T4 called tetraiodothyronine. That's why they call it T4. There's four iodines. See these four iodines? It's the real name is tetraiodothyronine, and it is amino acid derivative. It's a tyrosine derivative. However, it is lipophilic. It's lipophilic. The rest of the amino acid derivatives are hydrophilic. All right, so as a rule, amino acid derivatives are hydrophilic. The exception is T4, that's lipophilic. All right. Now, you can see that these amino acid derivatives are relatively small because, I mean, we're going to be talking about protein hormones, which are hundreds to several hundred amino acids. This is the size of one amino acid. So these are relatively small, and they're hormones. They're hydrophilic hormones. There's lots of them. There's only three shown here, but there's really lots of them. All right. So these are amino acid derivative hormones. We have peptide hormones. Now these peptide hormones are short polypeptides or small proteins. Just so you know, some hormones, protein hormones, are going to have quaternary structure and have subunits of polypeptides. So it'll have like an alpha chain polypeptide and a beta chain polypeptide put together to make the functional hormone. But right here we're talking about the small proteins, the short polypeptides. These are called peptide hormones. Sometimes they're glycohormones. That means a carbohydrate moiety. Let me put that word here for you. A glycohormone, it means that a carb moiety, it's a fancy word for part, piece, part, is attached. A carb moiety is attached and it becomes a glycoprotein. Some examples of those are things like luteinizing hormone, thyroid stimulating hormone, follicle stimulating hormone, and in lab, I make you start memorizing this. The next lab you have, you'll be memorizing the abbreviations for all these hormones. You're going to have to. It's going to be a lot of memorization, but you got to do it. So sometimes these are glycoproteins. All right, so that's peptide hormones, and these are hydrophilic. These peptide hormones are hydrophilic. By the way, I just saw the word. I better explain this. Many times these are made as prohormones, inactive forms that are converted to the active form at some point in time. Sometimes they're made as pro hormones, and then they're converted to prohormones. Usually a cleavage occurs. Usually this is a cleavage of some part. And then the prohormone is converted to the hormone, and usually another cleavage occurs. Matter of fact, with insulin, that occurs. This used to be connected. All of this used to be connected from here to here, and from here to here. Underneath, say it goes. This used to be one big polypeptide, and there were some cleavage events that occurred. And actually, the peptide that was cleaved off is called the C peptide. That's what it's called. And wow, I can't erase those. Whoops, I'm sorry. I'm trying to erase these for you. It is not letting me erase them. You rotten thing. All right. Anyway, so insulin is made as this prohormone, and then when you cleave off a piece of it, what's left is this hormone right here. Now, what you might say is, what's the why? You might say, what's the function of that? And the answer is you can make as much as the prohormone as you want, and it's not going to have physiological activity until the cleavage occurs. So it allows you to pre-make the hormones without causing physiological changes, and then have a lot ready to go when you need it. All right, so that's peptide hormones. Then we have lipid derivatives, and there's two big classes of lipid derivatives. We have the eicosanoids. These are things like prostaglandins and leukotrienes. You hear about them on TV all the time because they have different drugs out there, non-steroidal anti-inflammatory drugs like aspirin and other things, and Singulair, and they say inhibits leukotrienes, or inhibits, usually with aspirin they don't tell you it inhibits prostaglandins, but it does. So we have these eicosanoids, and then we have steroid hormones. All steroid hormones have this four-ring structure right here. And where I just circle, I'm looking right here. These four rings. These four rings are actually called, just so you know, cyclopentanoephenanthrene. And repeating, so what's different if all steroid molecules, I mean, I'm not even talking they don't even have to be hormones. There are steroid molecules that aren't hormones. In fact, cholesterol is a steroid. Just so you know, cholesterol, which is not a hormone, is a steroid. Cholesterol has this four-ring structure. The four-ring structure is called, whoops, oh wow, and my eraser's not working for some reason. I wonder why my eraser's not working. Press the button and see. You rotten thing. Sorry about this, folks. My eraser's not working. So the four-ring structure is called cyclopentanoephenanthrene. What changes then is the side groups that you put on it. Like if you put an OH here or an OH there, or other side groups. There's tons of side groups that are put on. That's what changes with the steroid molecule. These are all lipophilic or hydrophobic. So if I say lipophilic, fat-loving, lipid-loving, by definition, that's hydrophobic because fats are hydrophobic. If I say lipophobic, let me go back. I don't want to write that on that page. If I say lipophobic, that's hydrophilic. Just so you realize the words. All right, so that's the lipid derivatives. Now, what's the big difference between them? Well, the water-soluble hydrophilic hormones can float around all by themselves. I mean, they're water-soluble. They can float around in the plasma of the blood all by themselves. They don't need to be bound to anything else to make themselves soluble. So they can free float. And they can be secreted by the endocrine cell here and float free floating through the plasma. They can bind their receptor on a cell, some distant site, and elicit the response. This cell will now do what it's told because the hormone bound to the receptor. The response is elicited. But look at the lipid-soluble, the lipophilic hormones. They also are secreted into the bloodstream, but they have to bind carrier proteins. They are not water-soluble. They would have a hard time floating around the bloodstream, which is predominantly water. So they have to bind these carrier proteins, which carry the hormone to its target cell. It does release the hormone in its target cell. You might notice something else. The water-soluble hormone didn't enter the cell. It bound to a receptor on the surface. But the lipid-soluble hormone actually entered the cell, and it bound to an intracellular receptor, sometimes called a nuclear receptor. And that's also another big difference between these. Not just the carrier difference, where water-soluble hormones can float by themselves, but lipid-soluble hormones have to have a carrier protein. But also that water-soluble hormones bind cell membrane receptors, and lipid-soluble hormones enter the cell and bind internal receptors. So that's two big differences between those hormones. So if I look at hormones and hormone receptors, well, I have the hormone here, and I can have membrane-bound receptors that the hormone binds to, or I could have internal receptors, nuclear receptors, that the hormone binds to. If it's a membrane-bound receptor, it somehow has to alter the cell activity. And what it does is it either activates G proteins, which you already learned, or it activates intracellular enzymes. You're going to say, well, G proteins alter intracellular enzymes. Yes, they do. So ultimately, you want to activate intracellular enzymes or open channels or something like that. You might do it through a G protein, or you might do it in another way. And then you have your active enzymes, and the cells respond. What do the nuclear receptors do? Typically speaking, the steroid hormone nuclear receptor complex turns on genes. That's typically what it does. Turns on genes, you have transcription and translation, you make new proteins. This is the blown up picture of a water-soluble hormone binding its receptor, right there. Now look at this. Here's what this picture is showing you. This blue hormone is also water-soluble. And if it left the bloodstream right here, it's not going to affect this cell. And the question is why? And the answer is the cell doesn't have a receptor for that hormone, hormone number 2. So see, hormones can be cell specific because the cell might not have a receptor for that hormone. So does every hormone affect every cell? No, it doesn't. Matter of fact, the hormone that affects most cells in the body is tetroiodothyronine, T4. But other hormones are really specific for what cells they affect. And how can that be? Because the cell's only affected by the hormone if the receptor is there. And it's not affected by the hormone if the receptor is not there. This is showing you a lipid-soluble hormone like the steroids, or the one exception in the amino acid derivative class, thyroid hormone, that also enters the cell and binds intracellular receptors or nuclear receptors. And these typically turn on genes of the DNA. All right, G proteins, as you know. When a ligand binds a receptor and activates a G protein, that G protein now turns on enzymes. Typically, it turns on guanylate cyclase or adenylate cyclase. And guanylate cyclase makes cyclic AMP, a real famous second messenger. Guanylate cyclase leads to the production of cyclic GMP, another pretty famous second messenger, although not as ubiquitous, not found in as many cells as the cyclic AMP. So G proteins quite often turn on adenylate cyclase and or guanylate cyclase and lead to second messengers, cyclic AMP or cyclic GMP. And then these turn around and elicit the cellular response. Okay, so that's a G protein mechanism. And this is showing you which hormones use the G proteins. We can see a peptide hormone. No, it actually doesn't give you an example. Yes, it does. Down here. Down here it says, okay, who uses G protein and cyclic AMP? And the answer is calcitonin does, PTH does. PTH is parathyroid hormone. Antidiuretic hormone does, adrenocorticotropic hormone does, follicle stimulating hormone does, luteinizing hormone does, thyroid stimulating hormone does. All of these use G protein and cyclic AMP as a second messenger. All right. Now, could you have a hormone that binds the receptor and activates a G protein that doesn't turn on adenylate cyclase but turns on another enzyme? Yes. In fact, we turn on phosphodiesterase sometimes. Now, you didn't turn on adenylate cyclase, you turned on phosphodiesterase. What does that enzyme do? Breaks down cyclic AMP. So you reduce the amount of cyclic AMP in the cell. And who does this? Epinephrine and norepinephrine can do this. Specifically when they're binding alpha-2 receptors. Remember back in the autonomic nervous system I said that the alpha-1 and the beta-1 receptors were excitatory, but the alpha-2 and the beta-2 receptors were inhibitory? Well, now you can see that how are they inhibitory? Turn on phosphodiesterase, break down cyclic AMP, reduce the enzyme activity, inhibitory. So that's G proteins and second messengers right there. This is G protein leading to a different second messenger called calcium. Yes, Calcium is a second messenger. So now we're not talking about cyclic AMP or cyclic GMP. Now we're talking about the hormone binding the receptor, activating a G protein and opening calcium channels. Calcium then floods the cell. Now, calcium is the second messenger, but in fact, calcium's not active all by itself. Calcium actually has to bind a protein called calmodulin. It's this calcium-calmodulin complex that turn around and activates a bunch of enzymes in the cell. But we still say calcium is the second messenger, even though it really does have to bind the calmodulin to elicit its effect. Now, who uses this mechanism? Oxytocin, releasing hormones from the hypothalamus, several of the eicosanoids. All right. So this is the calcium second messenger, still using G proteins.
The membrane-bound receptor has a receptor site for a small molecule, called a ligand, exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
Okay, so yes, you have to know this mechanism. So let's go back to the beginning here. First of all, when the ligand, or the hormone in this case, the hormone's a ligand, binds its receptor, you activate the G protein. The G protein is this alpha, beta, gamma. What does it mean you activate the G protein? GTP binds to it. In fact, that's why we call it a G protein, because GTP binds to it. So this GTP alpha, beta, gamma is now active. But really, the active moiety of it is the alpha GTP. That disassociates. So if you were ever to ask me, what specifically turns on adenylate cyclase? What specifically turns on guanylate cyclase? What specifically opens the calcium channels? My answer is going to be alpha GTP. It is this alpha GTP moiety that's the active part of the G protein. Well, how do you turn the whole thing off? Well, first of all, the ligand has to dissociate. Now I don't have an active receptor anymore. And in addition, the alpha subunit has an internal phosphorylase activity, which cleaves the terminal phosphate off GTP to make it GDP, right there. Alpha GDP is not active. And now the alpha GDP reassociates with the beta and gamma, and now it's ready for another cycle. And yes, you do need to know that mechanism. This is a collage for you, showing you all of the mechanisms right here. Okay. Let's look at the cyclic AMP second messenger and the calcium second messenger.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP within the cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated. Calcium ions also serve as second messengers. In response to a signal molecule from other cells, the cell surface receptor protein activates a G protein, which in turn activates the enzyme phospholipase C. This enzyme catalyzes the production of inositol triphosphate, which diffuses through the cytoplasm to the endoplasmic reticulum. The inositol triphosphate binds to and opens the calcium channels, allowing calcium ions to flow from the endoplasmic reticulum into the cytoplasm. The calcium ions trigger proteins sensitive to calcium to initiate a variety of activities.
Okay, a couple comments on that right there. When this phospholipase C causes inositol triphosphate to be made, this is showing you how it leads to calcium as a second messenger, and really the calcium calmodulin complex. But in the future of this lecture, of this chapter, I'm going to show you the inositol triphosphate diacylglycerol pathway. It's another second messenger, and it actually goes through this phospholipase C as well. So this movie really is packed full of a lot of foreshadowing to what's coming. For right now, let's just focus on the calcium as a second messenger. By the way, this inositol triphosphate, which is broken down, wait for the word, this inositol triphosphate, which is going to open the calcium channels, it can be broken down into, boy, it just left me. I hate to say this, but it just left me. Diacylglycerol and, holy cow, it's a diphosphate. Diacylglycerol and, I'm sorry, I can't remember it right now. But it's a diphosphate, an inositol diphosphate, but that's not the name of it. I forget the name right now. And those molecules themselves are also second messengers. And in fact, you've probably heard of lithium, how it treats bipolar disorder. Lithium acts on this inositol triphosphate pathway. That's how it elicits its effect. And we might talk about that a little later. We'll see if it comes up. Of course, there's lots of different directions you can go with mechanisms here and talking about therapeutic drugs, pharmaceuticals, and they act in these pathways. So, all right, it's calcium as a second messenger right there. All right.
When a ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
So this is huge that you understand the difference here. This is alpha GTP directly opening calcium channels. Alpha GTP, it didn't turn on phospholipase C, it directly opened calcium channels. Back here, you had the alpha GTP. It didn't show you the alpha GTP, it just says G protein. But remember, it's the alpha GTP turned on phospholipase C. Phospholipase C made ITP, inositol triphosphate, it's abbreviated ITP. ITP opened the calcium channels, and the calcium binds the calmodulin and does its job. So there's more than one way to get to this signal the end product of signal transduction. By the way, this whole thing is called signal transduction. It's how the signal from outside of the cell, the hormone, is transduced into the elicited effect inside the cell. It's called signal transduction. There are entire graduate courses just on signal transduction. That means a whole semester, all you talk about is how the ligand elicits the effect. And I'm thinking you're probably starting to see why there's entire courses called signal transduction, because it gets complicated. What's the power of this signal transduction? And the answer is biological amplification, signal amplification. One hormone can lead to this many activated protein kinases. How does it happen? Well, one hormone leads to, say, three active G proteins. Each of these, well, I wish it was easier math. I'm going to make it easier math. I'm going to make this 10, 11, 12. Each of these active G proteins, each one of them, turns around and activates four. I'm sorry, can't do math. There's 12 here. It should be four. Four adenylate cyclases. So this one goes down and activates four. This one goes down and activates the next four. 1, 2, 3, 4. And this one goes down and activates the next four. So say 3 active G proteins turn around and activate 4 adenylate cyclases apiece to give you a total of 12 active adenylate cyclases. Now each one of these adenylate cyclases makes a certain number of cyclic AMPs. I'm not going to do the math this time. But you can see now that's amplified into a greater number of cyclic AMPs. Each cyclic AMP activates more than one protein kinase, and of course the overall effect is one hormone leads to a whole bunch of active protein kinases. It's called signal amplification. That's the power. That's the power of this right here. All right.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP in turn will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
That signal amplification. That's the power of this. All right. Now, when a hormone binds its receptor, you normally have a normal background level of this hormone ligand, and of course it's going to its target cell, which has receptors. When a hormone binds its receptors, one of the way we control that signal output, that elicited response, is the cell containing the receptor will downregulate. Now, so if the hormone is in too high of a concentration for too long, it'll quit having its effect because the cells with the receptors will downregulate the receptors. They will endocytosize them, and they'll fuse with a lysosome, and they'll chop them up. Now the receptor is no longer on the cell. Now it doesn't matter how much the hormone is in the bloodstream, the receptors aren't going to receive them. This is called downregulation. Now think about this. Now obviously this is a water-soluble hormone and this is a cell membrane receptor, but this could be true for nuclear receptors as well. Now, so think about this. If someone takes anabolic steroids or too much of a particular hormone, why do you see the results you see, like testicular atrophy and maybe gynecomastia, men growing breasts? And the answer is because the receptors are downregulated. So now it doesn't matter how much of that hormone you have in your body, it's not going to have the same effect. And let's say it is anabolic steroids. You put a bunch of anabolic steroids in your body, your muscles grow big and you're happy. But now what happened is you downregulated those receptors. So now normal amounts of testosterone won't have the same effect, and it won't lead to the masculine secondary sex characteristics that you think of with men, like axillary hair, beards, genital hair, all that. And in fact, you'll have the opposite effect. You'll have some testicular atrophy, you will have some gynecomastia and things like this. So downregulation does occur. Just like downregulation occurs, upregulation occurs. Upregulation occurs when a cell makes more of the receptors. So more of the receptors are made, which makes this cell more sensitive to the hormone. And in fact, thyroxine does this for other hormones. So T4, thyroxine, can cause cells to upregulate receptors to make them more sensitive to other hormones. For example, T4 leads to the upregulation of epinephrine receptors. So epinephrine only has its maximal effect when you have a normal amount of T4. If you have hypothyroidism, epinephrine won't have its normal effect because there won't be as many receptors on the cells for epinephrine. So this can occur. You can have some hormones upregulate for other hormones. So just like we have downregulation, we also have upregulation. Uh-oh. My movie is not playing. Not good. All right, folks. I don't know why the movie's not playing, but for whatever reason, it's not. I will. Either post this separately. I will post this separately and get you the movie somehow. Sorry. Okay, now look at this. Hormones and nuclear receptors. Lipophilic hormones like steroids and eicosanoids, they are, first of all, they're lipophilic. They diffuse through the plasma membrane easily. And then they bind their internal receptor. So this is an intracellular receptor. Now look, it could be a cytoplasmic receptor or it could be a nuclear receptor. It doesn't really matter. I mean, yes, does it matter if you want to get into the nitty-gritty details? Yeah. But for us, it doesn't really matter. What matters is it's an intracellular receptor. Now, this hormone receptor complex turns on genes. In fact, this hormone receptor complex is usually a transcription factor. And these transcription factors, of course, turn on genes. And then you get your gene product, which is the protein. So this could be testosterone turning on your actin and myosin genes, making your muscles bigger. That's what this could be. But the bottom line is these intracellular receptors typically turn on genes. This is showing you, oh, by the way, you probably already know that thyroxine, T4, the thyroid hormone, increases metabolism. Well, what organelle is most directly related to metabolism? And the answer to that is mitochondria. What it's showing you is T4 enters the cell and binds an intracellular receptor, and that binds a receptor on the mitochondria, and that's activated to increase ATP production. So that's increasing ATP production. Meanwhile, the thyroxine receptor complex can also turn on genes, and that can help increase metabolism as well. So that's the remember, thyroxine or T4 is an amino acid derivative. Most amino acid derivatives are hydrophilic, lipophobic, but thyroxine is the exception to that. It's actually lipophilic, and being lipophilic, it enters the cell and binds internal receptors.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
Okay, I want to point something out before we move on. Remember that that lipophilic hormones. Right here, I'm looking at the steroid hormone protein carrier. Lipophilic hormones have to be carried in the bloodstream on a carrier. They can't float free. They don't dissolve in water. It's like oil and water, all right? So they can't float free, but if they're carried by a carrier protein, then they can be and the carrier protein itself is hydrophilic. Now they can be transported through the bloodstream. Well, let me tell you some steroid hormones. Cortisol is a steroid hormone. Aldosterone is a steroid hormone. Testosterone and estrogen are steroid hormones. So we have a whole bunch of these steroid hormones. You could actually make plenty of the steroid hormone and be deficient in the protein carrier and suffer from the disease hypocortisolism, hypoaldosteronism, hypotestosteronism, or maybe we would call that hypogonadism, whatever you want to call it if you're male or female. So we could suffer from the hypo condition of not having enough hormone. But the reality is you really make plenty of the hormone. You just don't make plenty of the carrier. So if you don't make the carrier, how can you transport the hormone to its target cell? You can't. So there are cases, some diseases, where you make plenty of the hormone. That's not the problem. The problem is you're not making plenty of the carrier. So we do need to understand these mechanisms so we understand what to address with certain diseases. All right, let's look at some endocrine reflexes. And I briefly talked about this on the first slide or first couple slides. So here's the basis of an endocrine reflex. Pituitary gland makes tropic hormones. This stands for releasing, or I'm sorry. No, it doesn't stand for releasing. It stands for stimulating. So tropic means stimulating. All right, so you could say thyrotropic hormone, or you could say thyroid stimulating hormone. Anyway, this tropic hormone goes down to its target endocrine cell and tells this target endocrine cell to make its hormone. And it does, and it makes its hormone, and its hormone goes to the bloodstream and hits its target cell and elicits its response. Meanwhile, some of those hormones from the target endocrine cell negatively feed back. It's a negative feedback and shut off the pituitary. So now the pituitary doesn't make as much tropic hormone. So now the target endocrine cell doesn't make as much of its hormone. And this is a self-regulating negative feedback system. This is how we maintain homeostasis. Now, it's also showing you, what's this red arrow? Well, remember that what's going on from the hypothalamus to the pituitary are releasing hormones. So I have a releasing hormone from the hypothalamus telling the pituitary to make the tropic hormone. The tropic hormone from the pituitary is telling the target endocrine cell to make its hormone. That hormone goes out to its target cell and says, do this. Meanwhile, the ultimate hormone here feeds back and shuts off the pituitary. Well, is it possible that it feeds back and shuts off the hypothalamus? Not only is it possible, it does. And that's why this red arrow is here. Because this hormone can also shut down the releasing hormone. So that's an endocrine reflex with its homeostatic feedback. Now, we're going to talk more about that reflex, but let me just tell you, there are actually 3 stimuli causing hormonal secretion. There's a humoral stimuli. That's a metabolite in the blood. A metabolite just means a chemical in the blood. So humoral stimulus is there's a metabolite in the blood that turns on the hormone secretion. There's neural stimuli. A neuron tells you to secrete the hormone. There's hormonal stimuli. stimuli. A hormone leads to an increase of another hormone. Hormone 1 leads to the increase of hormone 2. That's hormonal stimuli. So there's these three stimuli involved in that endocrine reflex. The first one is humoral stimuli. This picture's not in your book. But the humoral stimuli is this. What if my calcium is too low? Calcium is this chemical floating around the blood. What if I have hypocalcemia? That's what that would be called. My calcium is too low. Okay, no problem. My calcium is too low. My endocrine cell from my parathyroid gland, this is my parathyroid gland. My parathyroid gland detects that calcium is too low. It secretes PTH, parathyroid hormone, which turns on osteoclasts, which digest bone, which increase blood calcium. Okay? What if blood calcium is too high? This is my thyroid gland. Oh, this is not. Well, it could be, and I could talk about calcitonin. This is still my parathyroid gland. And now my parathyroid gland detects that calcium is too high. It reduces its PTH secretion. It doesn't secrete PTH. Therefore, I don't turn on osteoclasts. Therefore, I don't release calcium to the bloodstream, and I lower my calcium in the bloodstream. So that's a humoral stimulus. Okay, now, this is a neural stimulus, and by the way, you already know this. This could be a sympathetic nerve, and probably is. And it's following the adrenal medullary pathway, where the exception to the sympathetic neurons is that this preganglionic neuron is extremely long. All right, anyway, this could be a sympathetic neuron going down to the adrenal medulla. The adrenal medulla can secrete epinephrine or norepinephrine into the bloodstream. Now, I can have one neuron that comes down here and stimulates it. I can have another neuron that comes down here and inhibits it. Says don't secrete. So I actually can have dual control here. I can have one neuron that has a stimulatory effect and says, hey, release epinephrine. I can have another neuron that has an inhibitory effect and says, hey, don't release epinephrine. But either way, these are neural stimuli telling the endocrine cell to secrete or not secrete its hormone. And then I can have hormonal stimuli. And that's the classic example where releasing hormones from the hypothalamus turn on the pituitary. They secrete tropic hormones from the pituitary, go to the target endocrine cell, and they tell the target endocrine cell to make their hormone. The hormone goes out and has an effect. Meanwhile, this end hormone negatively feeds back on the pituitary, and negatively feeds back on the hypothalamus, and shuts down the releasing hormones and the tropic hormones. And this ultimately shuts down the production of this hormone right here. So it's a self-limiting negative feedback. And this is a hormonal stimuli, where releasing hormones stimulate tropic hormones, and tropic hormones stimulate end product hormones, and end product hormones elicit their effect and negatively feedback and shut things down. All right. Oh. One more thing I'd like to point out is this. Is sometimes the hypothalamus secretes inhibiting hormones. So I've been talking to you about the hypothalamus secreting releasing hormones. Like I said, there's a growth hormone releasing hormone. Well, guess what? There's a growth hormone inhibiting hormone. All right? So sometimes the hypothalamus secretes inhibiting hormones that inhibit the pituitary. And now you can see that not only is there this basic homeostasis negative feedback, but we can even fine tune it even further with inhibiting hormones from the hypothalamus. We really do fine-tune these. Now, the hypothalamus secretes a whole bunch of releasing hormones that go to the anterior pituitary. And then the anterior pituitary secretes all these tropic hormones. All right? But that's only the anterior pituitary. What about the posterior pituitary? Okay, no problem. The posterior pituitary stores two hormones, ADH and oxytocin. Those are the two hormones that the posterior pituitary stores. The posterior pituitary does not make these hormones. I don't know if you can hear me pounding on the desk when I'm saying that. You might hear that. I got to be clear. The posterior pituitary does not make antidiuretic hormone and oxytocin. Who makes them? Hypothalamus. Hypothalamus makes antidiuretic hormone and oxytocin, but they're stored in the posterior pituitary. And then when the hypothalamus wants the posterior pituitary to release them, it sends a nervous signal down here and says release them. And then they're released and they go do their job. We'll talk about their job as we come up to it. In addition, as you already know, the brain also can have a neural stimulus on the adrenal medulla. This is a sympathetic neuron right here. All right, goes all the way down to the adrenal medulla and says, hey, secrete epinephrine and norepinephrine. All right, so that's hypothalamic control over the pituitary and even the adrenal medulla. And that's the end of 18.2. Now, 18.2 is long, I get it, but the rest of the chapter is going to be me telling you about, talking about specific endocrine glands and specific hormones, and you're going to hear the same story over and over and over again. It's just going to be about specific hormones, but it's going to be one of those stimuli. It's going to be either a hormonal stimuli or a neural stimuli or a humoral stimuli, and I'm going to be talking about negative feedback and homeostasis. All right.